What we're going to learn today is about, the regulation of GIT by different hormones. And, the function of GIT is basically, secretion, isn't it? Secretion of enzymes, secretion of water, electrolytes, isn't it? Secretion of, and of course, secretion of hormones from the different hormonal cells in the GIT. Okay. And another thing is motility. Okay. So these are two functions: motility to, to move the chyme from, or the, digested material from one part of the GIT to another part of the GIT. And of course, the other, aspect that needs to be regulated is the sphincters. Okay, the opening and closing of sphincters. Okay. So these are, basically the, different functions of the GIT. Besides, of course, absorption, isn't it? Of course, that plays a very important role. But then absorption will be brought about by the process of digestion, by the process of, propelling the, the chyme from one part of the GIT to another part of GIT. Okay, so without having these, functions of motility and secretion, we would not be able to digest the material, food material, and probably will not be able to even absorb the nutrients, because the nutrients needs to be, the food needs to be digested into simpler forms. Okay, that is basically the function of the GIT. And another thing is that motility helps also in the, the removal of waste, whatever is, undigested. All right, whatever food material that is undigested, will be excreted as waste. And for excretion also you require motility, and then the control of secretion of water and electrolytes. Okay, so when we talk of hormones regulating GIT, it refers to that, hormones also plays an important role in controlling the secretion and motility of the GIT. Understand? So that is the overall. When you generalize, the function of the hormones, that is basically, the two functions. GI hormones constitute a group of hormones secreted by enteroendocrine cells in the stomach, pancreas, small intestine, and controls various functions of digestive organs. All right. So anyway, they can exert autocrine, paracrine function, or neuroendocrine function, as you can see here. Okay. Some of the hormones like gastrin, CCK, secretin, GIP, all right, gastric inhibitory peptide. Okay, their more of action is by what we call as the endocrine pathway. All right, or hormone secreting at one point of the GIT and acting on the other point of the GIT, and hormones secrete into the blood. Okay, so all these hormones, gastrin, CCK, secretin, GIP, they secreted in some part of the GIT and then scattered by the blood and then acts on the distant cell. Okay, so it has an endocrine mechanism of action. Then some of the cells are having paracrine action, and reputed for this is the somatostatin, secreted from D cell, acts on the G cells, neighboring cell, okay, in the stomach. And of course, somatostatin is secreted from different parts of the body. Even the brain secretes somatostatin in the form of growth hormone, inhibitory hormone. All right, and in the pancreas, and we'll come back to this when we discuss somatostatin. Then of course some of the hormones are communicated neuroendocrine, right, in a neuroendocrine fashion. Okay, so some of these hormones are somatostatin, VIP, and GRP. Okay, so others are like acetylcholine, norepinephrine. Okay, these are also hormones that behaves in a similar fashion. Okay, so let's come back to this, and let's see the classification of GI hormones. You'll find that they can be divided into five family. One is the gastrin cholecystokinin family. All right, cholecystokinin is a bigger molecule than gastrin, but then it contains some gastrin within it. All right, component of gastrin within it. Okay. Then secretin family consisting of secretin itself, glucagon, vasoactive intestinal peptide, gastric inhibitory peptide. These are all secretin family, belonging to what we call as the secretin family. Secretin is the first hormone described, first hormone discovered by Starling and Bayliss. All right, the first hormone that was ever discovered. All right, among the hormones. Somatostatin family, then motilin family, and the substance P. All right. So these are, the different GI hormones. And we come to the gastrin first. Okay, this is a peptide hormone. Most of these are peptide hormones. Rather, all of them peptide hormones. Okay, and gastrin is one of them. Gastrin is a peptide hormone, and it depends. It can be a hormone containing 14 polypeptide containing 14 amino acids, or it may contain 17 amino acids, or it may contain 34 amino acids, or 43. So depending on the number of amino acids, it's given different names. So G, for example, G14 is called as a mini gastrin. Okay, and G42, if I'm not mistaken, is called as a big gastrin. Okay. So there are different forms of gastrin, and the fact that we have all these different forms of gastrin containing, where there is a variation in number of amino acids, such a phenomenon is said to be, it's called as macro heterogeneity. Geneity, all right, heterogeneity. Okay, that means heterogeneous. All right, comes from the word heterogeneous. So there is something called as a macro heterogeneity in the forms of gastrin that is being secreted. Okay, and some book have also mentioned about something called as micro heterogeneity. Okay, where probably a single amino acid is being either sulfated, or there's some kind of difference in one amino acid in the different forms of gastrin. Anyway, this hormone is released by G cells, and G cells are present in the what we call as the pyloric antrum of the stomach. They're also secreted from G cells in the duodenum, G cells in the pancreas. All right, and G cells are cells that secretes the gastrin, of course, and then that has a receptor that we call as the cholecystokinin B receptors. The cell in the stomach, they have this receptor. All right, and where the gastrin secrete from the G cell will bind to this to stimulate the release of histamine, and histamine is secreted by the enterochromaffin-like cells. All right, so G cell binds to enterochromaffin-like cells. All right, there are receptors on the enterochromaffin-like cells for gastrin, which are called as cholecystokinin B receptors, and this binding result in the release of histamine. Histamine acts on the parietal cell, and parietal cell, the parietal cell is stimulated, and there will be secretion of acid from the parietal cell. That is one thing. The formation of HCl, but then what is important is the insertion of the proton pump. All right, hydrogen potassium ATPase pump. Okay, so the insertion of this is a result of the stimulation of the parietal cell. So parietal cell can be activated by histamine, or rather gastrin through histamine, or you can also have a direct stimulation of the parietal cell by G cell, by the gastrin itself. Okay, so you have receptors on the parietal cell also for gastrin. Okay, so you can have a direct or an indirect activation of the parietal cell, and this result in insertion of hydrogen potassium ATPase pump results in the secretion of hydrogen, and which then mixes with chloride to form HCl. And so HCl, as you know, is important for the digestion of food that is coming from the mouth into the stomach, and it helps also in the protection of the GIT. It's kind of an innate immunity where it kills the pathogens. Okay, acidic nature of it kills the pathogen. But then you will find something that is paradoxical, that if the stomach is highly acidic, then it would prevent the release of gastrin. Okay, so anything, and then acidity, if the acidic environment in the GIT, in the stomach is of a pH that is less than 2, then you'd find that this would inhibit the release of gastrin. Okay, so what are the regulating factors of gastrin secretion? One is that it increases, one factor is that increases the secretion, and another factor is that decreases the gastrin secretion. Okay, so those factors increasing gastrin secretion are peptide themselves. So protein that is broken down into peptides, the amino acid themselves, phenylalanine, tryptophan. These are very strong amino acids that can stimulate the G cell and potentiate the secretion of gastrin. Okay, then you have of course a neural control, vagal stimulation via the GRP. All right, and for that reason it cannot be blocked by atropine because it's not via acetylcholine. Okay, then other things like calcium also have a role in increasing the secretion of acid from the secretion of gastric juice, all right, or by increasing the gastrin secretion, all right, from G cells. Okay, so factors decreasing gastrin secretion, one is acid itself. As I've said, if the the chyme, or sorry, the what we call it, the acid pepsid chyme, all right, that is present in the stomach, if that, which is a digested basically digested food mixed with pepsin and acid, if the acidity of that is less than, pH is less than 2, then that would cause a kind of a negative feedback and decreases the secretion of gastrin from G cells. Okay, then of course you have stomatostatin. We'll come back to this. This is a hormone that inhibits almost secretion of all the hormones in the GIT. Okay, and of course other hormones like secretin, GIP, VIP, glucagon, calcitonin, these also plays a role in decreasing the the secretion of gastrin from the G cell. Okay, so when you look at G cell, you'll find that it basically controls the secretion of hydrochloric acid, pepsinogen, intrinsic factor. So that means it plays a very important role in digestion, in protecting the GIT, all right, and of course in the absorption of vitamin B12. Okay, so that is one thing. But then gastrin is not only, the function of gastrin is not only restricted to secretion alone, but then it also helps in motility of the stomach. All right, not only the stomach, but even the duodenum and the small intestine and the large intestine. So these are also, their motility can also be controlled by gastrin. So motility is one function, secretion is another function, but then you have other functions also of gastrin. For example, it can also help in the release of insulin. Okay, so but then not in a physiological dose, in a very small dose. There is evidence that gastrin do increase, rather do help in the secretion of insulin from the pancreas. Okay, then it potentiates the the secretion of glucagon as well, all right, calcitonin. Okay, so these are some of the other functions of gastrin, all right, and if you go into your textbook, probably you'll find one or two more. Okay, but these are basically the functions of gastrin. Okay, then CCK, which is again a hormone from the same family. So these two hormones, gastrin and CCK, they belong to the same family, group of hormones. Okay, and again it is composed of varying numbers of amino acids, and you have different forms of CCK. Again, can be CCK8, CCK22, CCK33, CCK38. I think around 4 or 5 types are there. Okay, and they are secreted in response to a meal. They are basically secreted in response to peptide, all right, and fat. These two things, fat and peptide. It's and CCK plays an important role in the digestion of fat and protein. Okay, and so as I've said earlier that every form of CCK, it may be CCK8, CCK22, CCK33, but then they will have same 5 amino acids at C terminal as gastrin. So there is a component of gastrin in CCK. Okay, so that's why they are said to be clubbed in the same group or same family. Okay, because of this feature. And action of CCK basically to cause contraction of gallbladder, all right, where the bile is being stored. Okay, so that the bile can be secreted into the intestine, the gallbladder needs to be contracted, and so that the bile can flow into the intestine, the sphincter of OD needs to be relaxed. So there is a concomitant action on these two: smooth muscle that contracts the gallbladder and smooth muscle that relaxes the sphincter of OD. Okay, so I mean this helps in the flow of bile from the gallbladder into the intestine. So that is one thing. And as you know, bile is important in the digestion of fat. Okay, emulsification and exposure of that helps in the exposure of the fat to different enzymes. Okay, so we'll come back to this when we discuss the fat digestion. But anyway, it's important for fat. So even fat itself is a stimulus that activate the secretion of CCK, and then protein as well, the peptides. Okay, the digested product of protein. Okay, peptides, amino acids. All right, these also have a role in increasing the secretion of CCK from the cells in the that are present in the duodenum and jejunum. All right, these are called as the K cells. Okay, so these are cells that secretes the CCK. Okay, K or I cell. I'm not very. I think one of them, K or I cell. Okay, so anyway, these cells are present in the duodenum and jejunum. That is for sure. Okay, and when fat and digested protein arrives in the duodenum and jejunum, these cells will respond by secreting CCK, and CCK will help in the secretion of bile, number one, and number two, it helps in the secretion of pancreatic enzymes. Okay, so from the exocrine pancreas, these enzymes will be secreted, and they'll help in the digestion of protein. You will be reading about when we go into exocrine pancreas about trypsin, chymotrypsin, all those carboxypeptidase, all right, neuropeptidase, all right, and different enzymes that help in the digestion of protein. Okay, so number three is that it augments the secretion of secretin. You see, in secreting the enzyme from the pancreas, you need to have some kind of fluid along that that can be that can flow along with the enzyme, so that the enzyme can be carried from the pancreas into the intestine. Okay, so you need a medium, and this medium is being secreted by the the secretion of this medium is helped, it's actually controlled by secretin. So it'll be in the interest of the function of CCK that it also augment the effect, its action rather, augment the action of secretin. Okay, so secretin will be helpful in producing an alkaline type of pancreatic juice. Okay, the secretion of an alkaline type of pancreatic juice. All right, these are all with respect to secretion, but then it also plays an important role in motility by decreasing in gastric emptying. Okay, so when you say decreasing gastric emptying, that means it's going to decrease in gastric motility, one, and two, it is going to control the sphincter, the pyloric sphincter, and close the pyloric sphincter. Okay, so that is another function. Then another function is that induce satiety by acting through hypothalamus. So it has a role in in the control of feeding as well, feeding and satiety. Okay, so it is factor controlling, producing satiety. That means a person, stops eating food. When the fat and protein arrives in the duodenum, CCK is secreted and then it acts on the hypothalamus. Then that, it acts like a kind of feedback mechanism, which is negative in nature, a negative feedback mechanism that, decreases feeding, all right, stops feeding. Okay. So what is the mechanism of action of CCK? It acts through, when it binds to the cell, then it causes activation of phospholipase C, another enzyme which is present in the cell membrane. And what happens when this is activated? It causes the breaking of what we call as phosphoinositol diphosphate into inositol triphosphate and diacylglycerol, and this would increase calcium concentration within the cell and activate protein kinase, and then result in the release of these granules from the pancreatic cells. All right, so pancreatic cell. This is how it acts in the pancreas. Okay, this is how CCK acts on the cells in the pancreas to eventually result in the secretion of enzymes, okay, from the pancreas. So CCK may also involve stimulation of vagal nerves, all right, and then the intrapancreatic neurons which then stimulate the pancreatic acinar cell, and acinar cells are laden with these are granules containing pancreatic enzyme, eventually leading to pancreatic enzyme secretion. Okay, that mechanism, now this is a mechanism that you see occurring for hormones that are peptide hormones. Okay, steroid hormones would not act on them, would not bind to receptors on the membrane. They would directly diffuse through the cell membrane and then act on receptors present in the cytosol. But this action itself says that this is a peptide hormone. Okay, so controlling factors of CCK anyway is secreted from duodenal mucosa and jejunum also, all right, from the duodenum and jejunum, and monitor peptide by pancreatic acinar cell, secreted in response to fat protein digested products, digested proteins, I would say, all right. And of course, it also is controlled by the neural inputs that you get in the cephalic phase of CCK secretion. Okay, these peptides are degraded by pancreatic trypsin. Anyway, that I think will not go into this. I have already spoken all that is written here. Okay, so next next hormone would be secretin. Okay. One thing more about CCK is that even CCK has a role in increasing helping with the secretion of insulin. Okay, just like gastrin, but not in a physiological dose. Okay, then secretin is another hormone. Now this is, we don't have that the different forms of secretin. There is only one form, and it is a polypeptide of 27 amino acid. Okay, and it's secreted by S cells located deeply in the mucosal gland of duodenum and jejunum again. Okay, this is therefore easy to remember. Secretin and CCK, they are the cells that secrete, these two hormones are present in the duodenum and the jejunum. Okay, and secretin's structure, chemical structure is similar to glucagon, BIPs, GIP. That's why they are, clubbed in the same family. So only one form has been isolated. The first hormones to have been, found, discovered, and isolated. Okay, and it's usually stored in an inactive form as prosecretin. Okay, then when it gets, then only when it's released into the intestine it becomes activated into secretin. Okay, so what stimulates this? The secretion of secretin. As you know, secretin is important in the release of an alkaline juice, all right, from the pancreas, from the liver as well. Okay, so a rich, fluid containing, which is rich in bicarbonate, we can say. Okay, fluid which is rich in bicarbonate. So that it secretes, that it helps in secretion of this kind of fluid itself says that secretin helps in reducing the acidity, all right, of the chyme that is arriving in duodenum and jejunum. So what stimulates secretin? it will be the acidity in the of the chyme. All right, acidity of the chyme that is reaching the duodenum. Okay, so the acid chyme is therefore the most important stimulant of the S cells in the duodenum and jejunum that results in secretion of secretin. Fine. So what is the action of secretin? As I've said, most potent humoral stimulator, that means hormonal stimulator of fluid and bicarbonate secretion by pancreas, by the liver as well. All right. So what does it do is that it causes the ductular cell which are present in the liver, the liver, the gallbladder, the ducts going into the gallbladder and out of the gallbladder, and then the pancreatic ducts, okay, and also the centroacinar cells in the pancreas, okay. It causes the secretion of bicarbonate from these ductular cells and centroacinar cells, all right, eventually resulting in the secretion of a fluid rich in bicarbonate, which is therefore alkaline in nature. Okay, so an alkaline fluid. Okay, and this is important to neutralize the acid that is present in the acid chyme. Okay, which is arriving in the duodenum and jejunum. Okay, so it acts in concert with CCK. This is important. Why? Because this rich alkaline fluid is coming from the pancreas. Is to also not only target the acid chyme of neutralizing it, but then also acts as a kind of a medium because it's a fluid. All right, water, which is just water and which is rich in bicarbonate. So this water also helps to form a kind of medium for the secretion of enzyme from the exocrine pancreas. Okay, so chymotrypsin, trypsin, all these enzymes, carboxypeptidase, nucleopeptidase, all these are carried by this fluid, and then can be secreted into the duodenum and into the small intestine. Okay, and so it does two things. It helps in the secretion of enzymes, all right, which is stimulated by CCK, all right, but then together with CCK it acts in concert, all right, for the secretion of these enzymes. Okay, and then what does it do? Helps in the neutralization of the acid chyme, and of course digestion of the protein and fat. Protein, fat, and carbohydrate as well. Okay, because you get all those enzymes present in the pancreatic juice. Then it acts through cyclic AMP, the mode of action. You have seen that CCK acts through phospholipase C, all right, and you have second messengers as IP3 and DAG, but here it was going to be cyclic AMP. That means it's going to stimulate the adenyl cyclase, isn't it? And adenyl cyclase is going to convert ATP into cyclic AMP. Okay, and then cyclic AMP is going to result in the production of the phosphorylation of downstream enzyme like protein kinase A. All right, and all right, let's see. I think here we have this mechanism of action of different hormones. Here CCK, all right, acts on this. CCK binds to a receptor as G proteins are activated, and phospholipase C is activated, all right, and phosphoinositol or we call diphosphate is phosphorylated, and then resulting in the formation of inositol triphosphate and diacylglycerol that eventually activate the protein kinase C. Okay, and this altered phosphorylation of structural and regulatory proteins leads to vesicle insertion. So this vesicle is inserted here and then result in secretion of protein or enzymes. Okay, so in this case it will be enzymes, all the enzymes, pancreatic enzymes. Okay, but in case of secretin, it binds to these cells. In this case the cells would be ductular cells and the centroacinar cells in the pancreas, all right, and see again G proteins are stimulated, and then of course that results in the stimulation of adenyl cyclase, and finally the activation of protein kinase A. Again, this also helps in the insertion of these vesicles. Okay, and these are laden with not only enzyme but then helps in the secretion of a fluid that is rich in bicarbonate. Okay, fine. So same with a VIP, which we will be discussing later. Alright, so this is the mechanism of action, okay, of not only CCK but secretin as well. Okay, but secretin is through adenocyclase. CCK is through phospholipase C. Okay, so what is the action of secretin? Now we know it increases pancreatic enzyme secretion by augmenting the function of CCK. Alright, and at the same time it helps in the secretion of a fluid which is rich in bicarbonate, okay, and alkaline secretion. Number 2, it can decrease gastric acid secretion, okay, and as motility as well, okay, and pyloric sphincter contraction also is a function of secretin. As you know, and CCK and secretin therefore controls the overall function of gastric emptying. That means the contents from the stomach will be prevented from flowing into the duodenum. That is to put in a simple simple statement. Stimulate growth of exocrine pancreas. Okay, so they work in tandem with CCK. Okay, so these are all functions of secretin, and secretin also potentiate the release of insulin. Okay, so what controls secretin? The acidic chyme, number 1. Of course, protein digested products, number 2. And of course, these are factors that increases the secretion of secretin. But then you have somatostatin and metencaphalin, which inhibit the secretion of secretin from the S cells. Okay, so these are the controlling factors of secretin secretion. Then you have smaller, hormones like gastric inhibitory polypeptide. Now this is also a member of secretin family. All right, acts in the fashion of endocrine hormones. Now it's basically a polypeptide of 42 amino acid. I think we don't have to remember all these pro hormones and all that. Okay, but we have to remember, this is K cell. So the the CCK is released from the I cell. So GIP is released from the K cells, which are found in the duodenum and jejunum. So there's sometimes we get confused by the names of these cells. So K cells in the duodenum and jejunum releases GIP, whereas I cell in the duodenum and jejunum releases CCK. Okay. Now you have GIP receptors, which is a 7-transmembrane protein found in the beta cells of pancreas. See, beta cell is a cell that secretes insulin. Now GIP is the only hormone that has the ability to stimulate the beta cell, in the pancreas, and causes the secretion of insulin in a physiological dose. Others, all these other hormones, they have the ability of stimulating beta cell, but they cannot cause secretion of the insulin in a physiological dose. It's only gastric inhibitory polypeptide that has this role. So one thing is that we must understand is that GIP secretion is actually stimulated by carbohydrates, glucose, and fat. Okay, that itself gives us a clue that it has an important role to play in controlling the glucose metabolism. Okay, so this is otherwise also called as there is another name for this because it plays a role in the control of glucose metabolism. This hormone is called as an incretin. It's called as an incretin. Okay. Earlier they used to club all these hormones together with GIP, all secretin, CCK, gastrin, and they collectively call them as incretin. But actually, the only hormone, GIT hormone, which has the ability to cause secretion of insulin in a physiological dose is GIP. Okay. The rest would cause secretion, but in a very, very, very small amount. Okay, it cause secretion of insulin, but in a very, very small amount. not in a physiological dose. Understand? So anyway, what are the actions of GIP? One, as I've said, yes, stimulated by glucose and fat, okay, and as well as by acid in the stomach. And one thing that it does is that it can cause decreased gastric motility, but this is a very mild, function. It has a very mild effect on this type of function, but it plays a very important role in inhibiting gastric acid secretion directly and indirectly, okay, by acting on the parietal cell itself, or by acting, or indirectly by inhibiting gastrin release from antral G cells via somatostatin, or by acting via somatostatin. So it can be a direct inhibition of parietal cell, or by stimulating D cell to release somatostatin, and then this will act on G cells. We inhibit the G cells. Okay, so this is how inhibition is being of gastric acid secretion is being conducted by the GIP. Then, as I've said, stimulate insulin release, all right, from the beta cell. Okay, and for this action, that is also called as a glucose-dependent insulinotropic peptide, or as I've said earlier, the incretin. Okay. Now you have another hormone which is called vasoactive intestinal peptide. This is again only one form. It is a 20 amino acid polypeptide. All right. It is released in response to esophageal and gastric distension, vagal stimulation, fatty acid, ethanol, that means alcohol in duodenum. All right. Amino acid and glucose don't affect VIP release. All right. And action of VIP, it induces smooth muscle relaxation. That means relaxation of the stomach, the gallbladder, that is being induced by the VIP. Stimulates secretion of water into pancreatic juice and bile. Now this is very important. Electrolytes and water is being, the secretion of electrolyte and water into the GIT is actually helped by this, by this hormone. So there are instances where you have a tumor of these cells secreting VIP, resulting in what we call as a vipoma. In such circumstances, a person would come with diarrhea, okay, because there would be hypersecretion of these the secretion of these the intestinal cell would result in a secretion of water, and high secretion of water, increased secretion of water and electrolytes, and that would result in diarrhea. Okay. Then it also inhibits gastric acid secretion and absorption of the intestinal lumen. So it also functions as in stimulating pepsinogen secretion by chief cells, and it plays an important role in the vasodilation, blood vessels, most importantly the coronary blood vessels around the heart. So coronary vasodilation, it plays an important role here. Okay, so these are all functions of VIP. Somatostatin is another hormone. And this hormone, as I've said, is basically playing an inhibitory role. It inhibits the release of all the hormones in the GIT. See, it inhibits secretion of gastrin, VIP, GIP, secretin, motilin, GH, insulin, glucagon, except CCK. All right, except CCK. All right, but then otherwise all hormones in the GIT are being inhibited by somatostatin. And I've said that this is a hormone that is similar to what we call the growth hormone, inhibitory hormone released from the hypothalamus. It's the same hormone, as a matter of fact. It was first found in hypothalamus, so that itself speaks volume. Secreted by D cells in the stomach, duodenum, pancreatic cell. So you have these also in the pancreatic cell, the D cells, right? You remember in the islets of Langerhans, I think we have not done the endocrine pancreas. So you just note this, all right, that you'll also find D cells in the islets of Langerhans, okay, in the pancreas. Now secrete a large amount into gastric lumen, and therefore into the circulation finally, and acts in an endocrine fashion. Okay. Now this somatostatin is released in response to acid in the stomach, but I think it also is a kind of there is a kind of feedback here. So it will also respond to the concentration of all these hormones, all right, and then only it will be released, because its role is to control, inhibitory, that is to control the concentration of all these hormones. Then other functions are that increase fluid absorption, and decreased secretion from intestine, decreased endocrine, exocrine, pancreatic secretion, decreased bile flow and gallbladder contraction, decreased, gastric acid secretion and motility, decreased absorption of glucose, amino acid, and triglyceride. It plays it's a major, factor of inhibition in almost all aspects, not only the secretion, but then even the, absorption, the contraction of the gallbladder, all right. So motility of the gastrointestinal tract. Okay, so it plays an important role in inhibiting most of the functions. Okay, yes, it's a hormone that is presented in two forms. You have somatostatin 14 and somatostatin 28, and this hormone is similar to, in action, all right, it's similar to secretin because it stimulates adenylyl cyclase, all right, resulting in the formation of a second messenger known as cyclic AMP. So the mode of action is similar to any peptide protein, a peptide hormones. Okay. Another hormone is called as motilin, all right, and this is the last hormone that I will be discussing. Polypeptides secreted by enterochromaffin-like cells and the Mo cells. These M cells are also called as Mo cells, MO cells. Okay, so these are two cells which are present in the stomach, small intestine, and colon, and they, when you stimulate by number of things, okay, by a stretch, you will find that these cells would be stimulated and then causes the release of or the secretion of motilin. And function of motilin is that causes contraction of smooth muscles in the stomach and intestine, increases, and these contraction increases during interdigestive state. Every 90 minutes, there would be a contraction that is being caused by this hormone motilin, all right, and it behaves like a hormone that function in house cleaning, all right, housekeeping rather, okay, by pushing all the the chyme, the digested matter from the stomach into the intestine and into small intestine and then into the large intestine. Okay, so this kind of movement that you see, which is caused by motilin every 90 minutes or in the range of say 90 to 120 minutes, okay, in a periodic manner, all right, it's also called as the migrating motor complexes. Okay, migrating motor complexes. So these are the migrating motor complexes. Every 80 to 120 minutes there would be contraction. In duodenum it is 3 to 6 centimeters per minute. In the ileum is 1 to 2 centimeters per minute. Okay, so this kind of contraction happens from the it starts from the antrum and then it doesn't go to the large intestine. It ends in the ileum, okay, before reaching the large intestine. Okay, so what organizes these migrating motor complexes? One, they have said that it's the enteric nervous system, which is important, all right, in the generation of the MMC. But then you have other hormones also that plays a role in the generation of MMC, which is motilin, and then you have hormones that inhibits the generation of MMC, which is CCK and gastrin. Okay, so motilin actually plays a very variety of function, and in the gallbladder it increased gallbladder emptying, in the brain it increased hunger, and pancreas increased insulin secretion, rectum increased rectal compliance, and the entire gastrointestinal tract cause inhibition of gastric accommodation, induction of gastric phase 3 contractions, increase lower esophageal sphincter pressure, okay, enhance gastric emptying. Okay, so these are all the different functions of motilin, but we must remember that motilin is always associated to the formation of migrating motor complexes, all right, which plays an important role in house cleaning, or rather in removing all the digested products, in the stomach and in the small intestines, and then push that all into the large intestine. Okay, so every 90 minutes this is occurring, every 90 minutes, all right, or somewhere in a range of 80 to 120 minutes. Okay, so and the purpose is to remove push the chyme down into the rectum, down into the large intestine. Okay, so these are in brief, in summary, summarization of all the digestive hormones in the GI tract. You can download these slides and go through all these slides, and then you'll find that this is one thing that I want to discuss. There is, it is not that hormones act, in isolation, but their action is actually integrated. Okay, as you can see here, gastrointestinal stomach stimulates the release of gastric juice, which contributes to production of chyme, and then chyme in the small intestine stimulates intestinal cells and mucosa to produce secretin and CCK. As you can see, gastrin helps in the production of acid chyme. Acid chyme in turn helps in the secretion of secretin and CCK, and then secretin, is going to, of course, neutralize the acid chyme, all right, acidity in the chyme, by releasing sodium and bicarbonate from the pancreas, exocrine pancreas. CCK, of course, is going to help in digestion of proteins, fat, and carbohydrate. Then sodium bicarbonate neutralizes acid in small intestine, of course. Then secretin increases rate of bile secretion in the liver, all right. So helps in the secretion of bile as well. Secretin, not only the secretion that is coming from the pancreas. Okay, and finally, CCK signal gallbladder to contract and pores contends into small intestine. Okay, so one thing that is happening helps in the secretion of gastrin, actually eventually helps in the secretion of other intestinal hormones as well. Okay, and so this is kind of an integrated function that is happening, or that is happening in the GIT, and all for controlling for digestion and for controlling the flow of the digested material from the stomach into the small intestine, from the small intestine into the large intestine. Okay, so it's all about the basic functions of the GIT. Okay, so I think that is all about the discussion on the hormonal control of GIT. Okay,